Okay, our final talk of this session is Ryan Gutenkunst. And while he comes up here, I'm going to remind you, not only do we have another session at 4 o'clock, but we will hear the winner of the Crow Award in tonight's in tonight's session. So, in the keynote 3. Yes.
7:25, so it's just before the session.
Okay, so I'm just going to repeat this. 7:25 for the big news.
So recently my group has been working on extending both the theory and the inference of distributions of fitness effects in natural populations. And today I want to tell you 2 stories. The first about the DFE between populations is very much work in progress, so I'm eager to get constructive criticism about our methods or the data we're applying it to. The second story is a published story, but I think it illustrates a really cool connection between population genomics and biochemistry. And so to begin with, many people in the room are probably familiar with the idea of a distribution of fitness effects. But just to sum up is that this distribution quantifies how likely it is that a new mutation arising in a population will have a given selection coefficient. And in general, the distribution is bimodal with some proportion of extremely deleterious or lethal mutations and some proportion of moderate effect mutations. So today we're going to be focusing on inferring the distribution from population genomic data. And in that case, we're really only sensitive to this moderate effect peak, which may include deleterious mutations and beneficial mutations and nearly neutral mutations. So the typical approach is to infer these sorts of distributions from distributions of allele frequencies. And because it's a population genetic inference, in general, we're inferring not the distribution of S, but the distribution of gamma, which is the selection coefficient scaled by the effective population size. And the very generic strategy for doing so is that we fit we take some class of putatively neutral sites in our data. We fit a model of demographic history to those data because that demographic history is going to affect variation of all types of sites in the genome. And then we take the class of sites that we're interested in and we fit a model of selection plus demography to that class of sites. So the new thing we're trying out in an ongoing project is to think about the distribution of fitness effects not within a single population, but between pairs of populations. Because if we consider 2 recently diverged populations, in many cases the same alleles will be segregating in the 2 populations. And because of environmental differences or differences in genomic background, those alleles may have different selective effects in the 2 populations. So instead of a single univariate distribution of fitness effects, we now have a joint distribution for the fitness effect S1 in population 1 and the fitness effect S2 in population 2. And we can imagine a few extreme situations. For example, if there were no relationship between fitness effects in the 2 populations, then we would have uncorrelated fitness effects, and the 2 S1 and S2 would be independent draws from some underlying distributions. On the other hand, if there were very similar fitness effects on mutations in the 2 populations, we would expect a very highly correlated distribution with all the density lying along the diagonal where S1 equals S2. And so in both projects I'll describe today, we've been modeling the joint distribution fitness effects as a bivariate log normal distribution. And then we have a correlation coefficient, rho, that quantifies how similar selection is in the 2 populations. So high rho, illustrated here, indicates that any given mutation will have very similar fitness effects in the 2 populations. So the data we'll be working with come from the Drosophila Genome Nexus, which was assembled by John Poole. And we'll be working with data from Raleigh, North Carolina populations and data from Chuck Langley's lab of Zambian populations or Drosophila, which are a reasonable proxy for the ancestral homeland of Drosophila melanogaster. So as a sort of data quality issue, just to mention that we took our allele frequency spectra generated from these data and we project down to a smaller sample size to minimize any errors that may come from data quality issues in calling rare alleles, and because some of our simulation studies suggest that our power doesn't depend that much on how big our sample size is. And so for the first stage of the inference, we take our synonymous, so we take SNPs and synonymous sites as our proxy for neutrally evolving sites, and we use those and data to fit a demographic model to our data. Now, I don't want to belabor this, but just to say that the demographic model matches sort of what we would expect, and that we have a large population size in Zambia, the putative ancestral homeland, and a very sharp bottleneck leading to the out of Africa event with gene flow between the North Carolina population and the African population. So having that demographic model in hand, then we turn to the non-synonymous variants where we expect to see stronger selective effects and use that to fit our joint DFE. So in this case, we now have a number of additional parameters. So we have mu-z and mu-r, which are the modes of the distribution of fitness effects, the marginal distributions in each population. We have sigma-z and sigma-r, which are measures of how wide the distribution is within each population. And then the parameter we care most about is that correlation coefficient. Because we're working with Drosophila, and there's lots of evidence of adaptive evolution in Drosophila, we also include terms in the model for positive selection. And so when we do our inference, we infer actually a very high correlation coefficient of about 0.97, suggesting that in general, alleles have very similar selective effects in the two populations. If it's strongly deleterious in one population, it'll be strongly deleterious in the other. If it's moderately deleterious, it'll be moderately deleterious in the other. Although looking at the residuals, comparing the model and the data, there are still some issues with fitting alleles that are private to one population or the other. So this is interesting, but maybe that one number is, it's hard to put that in perspective and to know what you expect biologically. So we actually think the more interesting analysis is to break our analysis apart and think about genes of different function. Because we might expect that as fruit flies have expanded to different environments, there are different traits that will be under more or less divergent selection in the two different populations. And so to address this question, what we've done is try to separate genes into different functional categories using the Gene Ontology. And then for each category, we take the data from those genes and keep the demographic model fixed, and then fit our distribution of fitness effects to just the SNPs from those genes. And when we do that, we see a wide range of correlation coefficients inferred, although in general, almost all positive, like we would expect. And again, I mentioned this is work in progress, and we're still trying to wrap our head around these results. But cherry picking, and I admit it's cherry picking a few examples, we can see, for example, if we focused on gene sets involved in various developmental processes, in central nervous system development, we see a very high correlation, suggesting that those SNPs have very similar effects in the two populations. Whereas if we look at development of the musculature, we see almost no correlation in fitness effects there, inferred there, which suggests that maybe differences in selective forces are stronger on traits affected by musculature development than traits affected by neuronal development. And this is an area where I think it's a really interesting analysis, but I would love to talk with some people who are better Drosophila experts than I am to think about how do we interpret these physiological results. So I mentioned this is ongoing work, and there still are methodological issues we have to address. So the first question is, we're assuming a log normal distribution for DFE, basically for mathematical convenience. It's not clear that that's the best distribution or the only distribution we should be fitting. The challenge is that simply there's been much less math done on bivariate distributions than there is on univariate distributions. For example, one might want to use a gamma distribution, but there are multiple ways of defining that in the bivariate case, and they have sort of inconsistent properties. We also need to do some more simulation studies and additional tests to understand how robust our analysis is to different sorts of errors in modeling, say the demography, or errors in the data, those sorts of questions. So now I want to turn to a related project where we're applying this idea of a joint distribution of fitness effects to a very different problem, but encountering some of the same ideas. And this is work carried out by an excellent graduate student in my group, Aaron Ragsdale. And here we're not thinking about the joint distribution of fitness effects between populations, but rather between alleles. And in fact, alleles at the same site. And to do this, we're going to look at the small fraction of mutations that you discover in any sequencing study that are not biallelic, but triallelic. So there are 2 derived alleles. And in particular, we're focusing on cases in protein coding regions where there are 2 derived amino acid changes. And the question we want to ask is, how correlated are their fitness effects? You might imagine an extreme case where all that mattered was you made a mutation at this location in the protein. And no matter what mutation you made, it had roughly the same fitness effect, and that would imply a very high correlation. On the other hand, maybe the identity of the mutation is really important, and that would imply a much lower correlation coefficient. And I don't want to belabor this point, but just to say that I'm lucky Aaron is an applied mathematics student, because in the process of doing this analysis, he had to develop a novel numerical solution to the triallelic diffusion equation, which turns out actually to be quite non-trivial, more challenging than his advisor led him to believe when he started the project. So we're going to go back to the same data set, and here we're focusing on just a single population, the Zambian population. And we take the bivariate SNPs, the normal ones people work with, and use those to fit the demographic model and the univariate log normal DFE. And then when we turn to the triallelic data, the only additional parameter we have is the correlation coefficient. And so our inference, if we take all the non-synonymous SNPs in the data, the non-synonymous triallelic SNPs in the data, we infer a correlation coefficient of about 0.5, which suggests that reality is somewhere in between in terms of identity versus location, the mutation mattering. And we can do the sanity checks you expect. If you look at derived alleles that are biochemically different, you get a much lower correlation coefficient. If you pick sites where the 2 derived alleles are very similar, you get a much higher correlation coefficient. But again, the single value is maybe something that's hard to wrap your head around. What I think is really cool about this project is that we can compare with biochemical experiments. And in particular, the data we're comparing with come from deep mutational scanning experiments where people use clever assays and high-throughput sequencing to assay the effects of tens or hundreds of thousands of mutations simultaneously at potentially every location within a protein. We saw an example of this last night in Applied to tRNA from George Zhang's lab. And here we see data from beta-lactamase in E. coli, and we can calculate that same correlation coefficient from the biochemical data, and we get quantitative agreement. And in fact, those 2 numbers agree within the error bars, and the error bars are larger on the biochemical side just because you were working one protein at a time. And interestingly, we can take data from proteins from E. coli, yeast, and humans, and we get the exact same correlation coefficient from all of them, which to me suggests that maybe what we stumbled upon is not something to do with Drosophila population genomics, but is actually a fundamental property of protein evolution. So just to sum up, we're trying to take this idea of a distribution of fitness effects and really push it in lots of directions to think about joint distributions of fitness effects. When we apply this idea between populations, we can see interesting differences between genes of different functional classes, which may be telling us how selection acts on different functions in different environments. And we're really looking forward to applying this to other population pairs in different species or with different divergence times or in different sort of environmental contrasts. In the published work, we've looked at within a protein to try and connect population genomic data to biochemical data, which I think is just super cool. And let me close by mentioning that Aaron had a poster at this meeting talking about his work on the 2-locus diffusion model, where we can start to think about correlations in selection coefficients between two different sites separated by some recombination distance along the chromosome. And let me last put in a plug that sadly for me, Aaron will be graduating in the winter. So if you're looking for an extremely computationally and mathematically skilled postdoc who doesn't mind getting his hands dirty with the data, talk to me or more importantly, talk to Aaron. And with that, I'm happy to address any questions.
Methodological question. Can you comment on the effect of draft? All of the methods assume independence between sites. What happens when you have a lot of positive selection?
Right. So yes, as your own work has shown, draft can distort the inference of the frequency spectrum. To be honest, we haven't looked at it in detail. Everything we're doing is sort of single locus right now. I think we have to do some simulations to try and dig out how much that's going to distort the signal.
So I thought it's a really nice idea to have this differential fitness effect analysis. And I had a question of how much data you need. You were splitting it into Go terms. And also, I wondered if you looked at non-coding, like conserved maybe functional non-coding sequence where possibly there might be more differential selection going on. And whether
Right. So we haven't done a super thorough power analysis. So the graph I showed does have so there are statistical error bars there that are reasonably small, so you can see the differences even with not. So they vary pretty widely, and I think actually for the biological interpretation, we'd be better off going with bigger sets of genes where we aren't trying to sort through 350 Go terms to find the interesting patterns. Sorry. Non-coding, yes. The second part of the question we have not looked yet. And I guess the challenge there will be finding ways to divide them up into interesting categories where we can really, so we're going to have to dig more into the annotations.
All right, let's thank all our speakers from this afternoon. You can catch them at the break for questions. And to remind you, back here at 7:25.